Breaking news live from Bozeman Central. Autotrophs have been found to use photosynthesis. Here to break down this groundbreaking discovery is C.B. Robertson, the famed cousin of a substitute teacher here in Palos Verdes. C.B., what do you think of this new development?
I'm out here exploring the Northern Californian wilderness. As you can see, these beautiful trees and bushes. I'm up here at my uncle's house. Here's my little cousin. And we're going to be showing you my amazing experiment that helped give the evidence for this discovery. I'm going to go inside in just a second and show you this amazing science. So Tommy, now I'm inside and I'm going to show you my awesome experiment to show you how these autotrophs, these plants, are able to use oxygen to do photosynthesis. Now what I'm going to do is I'm going to take this Elodea plant and put it in a pot of water and see what happens. As we're waiting for this Elodea to start making some oxygen, let me tell you a little bit about autotrophs. These are organisms that are actually able to make their own food using sources like sunlight, and they include plants, bacteria, cyanobacteria, and even some forms of algae. What they do is that they take this energy and store it in the chemical bonds, which they can then use later and break them down in processes such as cellular respiration to get energy. In plants, they use the carbon dioxide in the air, and through photosynthesis are able to make carbohydrates to do that job. And here's the proof of everything I've just said. As you can see, this Elodea plant is producing these bubbles, which are actually just oxygen. Look at the vigorous pace at which this oxygen is being released by this plant as it undergoes photosynthesis, and releases oxygen as a byproduct. As you can see, it is very quickly making energy for itself, as it has been cut off the stem, and because it knows this, it is making food very fast.
Well, CB, I'm not sure that the public even understands what you're talking about. So why don't we start with what a chloroplast is? And that's where I come in. Luckily, I've got my trusty whiteboard to help explain these concepts. To begin, why don't we start with something that you already know and love, the leaf. Now, the leaf is the simplest thing that we're going to be learning about, and it's also the simplest drawing. Now, let's get a little bit more complicated. Wow, that was cool. Now, why don't we zoom into this lovely picture so that you guys can understand it better. This here is called a leaf cross-section, and it's a pretty familiar picture, so you're going to want to get familiarized with it eventually. So we're going to begin with the cuticle, which is the outermost layer of the leaf cross-section. And below that, these two structures right here are the upper and lower epidermis. And in the middle, you have the mesophyll. And that is where these special little red dots are. And I'll explain what those are later, because they are the most important part. This right here is called the leaf vein, and the gases that are represented here are CO2 and O2. And CO2 enters the cross-section and O2 exits it. And here we have the stoma, which allows for the exchange of gases in the leaf. And now you might be wondering, what are these little red dots in the leaf cross-section? Well, if you guessed chloroplast, then you are right. So what are these mysterious red dot chloroplasts? Well, in real life, it turns out that they're not just as simple as being a red dot. They're actually quite complicated. And here I've drawn out a chloroplast. Basically, it's got an outer membrane, an inner membrane. It's got stacks of thylakoids, which become granum, and in the middle of all that is the stroma. Now, let's get even closer into one of those thylakoids and see what's really going on. And now we have reached the final level of complexity, looking at individual thylakoids. And now these thylakoids are made up of the thylakoid membrane, which is right here, and the thylakoid space, which is on the inside. Now, there are two different types that we've been learning about, independent and overlapping. Independent means that each thylakoid membrane is self-contained and they do not overlap, as opposed to here, where there is a gap in the membrane and the thylakoid spaces overlap, as seen here. Wow, those drawings were pretty cool. But now I think we should get into an animated short from our very own Ishan Gar.
Thanks, Tommy. I'll get right into it. First, we have the net equation of photosynthesis. 6 molecules of carbon dioxide plus 6 molecules of water to form C6H12O6 and 6 molecules of oxygen, which is a waste product. This is done using the energy from sunlight. Photosynthesis involves oxidation and reduction reactions, whereby the carbon dioxide is reduced and the water is oxidized, as shown in the equation. Next, let's compare the energy input and output of these light reactions. In goes ADP and NADP+, and using the excited electrons in the photosystems, these molecules are reduced and turned into ATP and NADPH. Also, remember, light reactions only take place in the presence of light. I mean, who knew? And they stored this energy temporarily in ATP and NADPH, changing the sunlight to chemical energy. Now let's talk about the Calvin cycle, the other component of this series of reactions. Melvin Calvin discovered it and won the Nobel Prize for it. Basically, the Calvin cycle reduces carbon dioxide to a carbohydrate in the stroma of chloroplasts. Let's look at the reaction. During the reaction, CO2 is taken up and then reduced to a carbohydrate that can later be converted into glucose, as seen in the diagram. One more thing, the reaction does not need light, like the light reactions do. Wow, that was cool. Back to you, Tommy.
Now, that's going to be all for Bozeman Central. Please like and subscribe if you liked what you saw, and shout out to our teachers, Ross and Mr. Robertson. Thanks, guys. See you later. Let's go.